Hey guys, just want to give you a heads up. There's about 5 resources that you should be using for AP Biology. First is the Google Classroom, first and foremost. That's where all your updates are going to be, and I really want you to be on Google Classroom as often as possible, hopefully daily. I'll be evaluating, reading, giving feedback, stuff like that. Let's chat it up. Second thing, with Google Classroom, you're going to have your chat challenges, and those are going to be designed to keep boosting your thoughtful processes. Then we're going to have, on top of that, you want to have your Pearson Campbell Brown workbook. You want to work through that. Now the third thing, I know that there's been a change in the AP exam, but that doesn't really help us per se, because I've always flipped the curriculum so that we can figure things out starting with an evolutionary and more macroevolution background. Most teachers don't do that because I don't know why they don't do it. I think it's the best way. But as a result, some of the things that they think we did in the beginning of the year, we haven't done yet. So really go on the College Board YouTube daily webcasts. I'll always have a link on the Google Classroom, but you want to check on those. You want to watch those. You don't have to watch them when they're streaming live. They're going to be archived, but you should watch them, have a notebook ready, pencil and paper, and be ready to work the problems. Fourth resource then is going to be any kind of supplemental things that end up on the Google Drive, homeworks, things like that. And then fifth, if you got your textbook, by all means, go through the chapters. I still would like to assign chapters on genetics and on molecular genetics and on biotechnology. So I know we can't get our hands on in the lab. There were like 6 really cool labs, and we'll actually have a couple weeks together to play in the lab. I don't know if that's actually going to happen, but I'm an optimist, so that's how I'm going to work it. So guys, stay tuned, stay on Google Classroom, chat it up, check your AP Bio folder on the Google document thing, but really Google Classroom is where everything's going to be alive and breathing. So I'll be interacting daily. We'll see you out there, chat it up, talk to you later. Talk to you real soon. Bye. Hey everybody, this is your AP Biology virtual handshake first assignment, and this is the key. So we're going to start off by looking at the idea of the overarching question. That's a question you don't want to answer right away. You got to wait for that one. Wait until you've done the supporting questions. The overarching question should come to you then. Here's the first problem. We're looking at the idea of a monohybrid cross. That means just one trait considered. In this case, a yellow pea versus a green pea. By trait, then that would be pea color. Remember, hetero means other. So heterozygous would be one yellow, one green allele, big A, little a. And we cross that, big A, little a, with big A, little a. Remember that the big A is an allele found on one sex cell, and the little a is an allele found on the other. And when we bring the two together, we're fertilizing those eggs. So we end up with 1/4 big A, 2/4 big A, little a, 1/4 little a, little a. That gives us 3/4 yellow under the rule of dominance, and 1/4 green. So that's number 1 on the supporting questions. We'll move on to number 2 now, where we consider a completely different rule, incomplete dominance. And that's where you basically are going to mix 2 colors, kind of like paint. So you can think of the I in incomplete dominance as our little mixing sticks here. And so we have red, pink, and white are your possibilities for the alleles. And remember, pink is from mixing a red with a white. So if I was going to take all pink snapdragons and breed them, what I'm going to see is big R, R prime, or pink snapdragons. That gives me an R and an R prime allele to deal with. When I make the Punnett square for my pink snapdragons, I'm going to end up with this little gem of a thing. I'm going to have big R, big R, big R, R prime, that's a pink one, big R, R prime, that's a pink one, and R prime, R prime. So I got a red, 2 pink, and a white. So RR is simply going to be red. That's 1/4. R prime is going to be 2/4, and that's where they mix like paint. That's my original pink snapdragon. And then R prime, that's that sort of really recessive thing. That's the 1/4 that are white. Now remember, I said recessive, but these are all incomplete dominants. They all have equality. When you're all done, this is kind of what you look at. You've got 1/4 red, 2/4 pink, and 1/4 white. All right, so that's question number 2. The next one looks at a heterozygous dihybrid cross. Heterozygous, both kinds. Dihybrid, 2 traits considered. And it doesn't really matter what our traits are. This kind of looks at the mathematical poetry, the waltz we talked about in the first week of the trimester. So I've got my big A little a, big B, little b, and I'm looking for all the possibilities, and I need one A and one B, one A and one B, one A and one B. And these are the 4 possibilities, right? It just matches for the other one. So big A with a big B, big A with a little b, little a with a big B, little a with a little b. That really is just emulating the rule of distributive property, right? This is just mathematics, a binomial. We've got distributive property going on here. You guys know more about math than I do. I just know more biology than most people I know. So correct me if I'm wrong, but this is the distributive property. All right, so once we have that distributive property, we can build our Punnett square, and we're going to get our dominant and our dominant with recessive, and our recessive with the dominant, and our recessive with the recessive, and we'll lay this thing out. We get 16 squares for a dihybrid cross, and you should never have to do a dihybrid cross again if you think about the mathematics of this. So, we're just going to bring our gametes together and build our alleles. 1A and 1B gamete with 1A and 1B gamete gives us a diploid, big A, big B, big B, and we just keep going through the zooming through the Punnett square here. And if you look, I've got those 4 in a row. That's a really nice pattern. Then 2 in a row. That's a nice pattern. 2 in a row. That's a nice pattern. And there is a hidden 2 in a row right over here. There's a little gap in there. I call that hopscotch. And there's another little 2 in a row right here. And that's another hopscotch. And then I've got a couple of singles. So you can see then down from left down to right, there's a nice single, 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 single. And from left going up to right, there's that nice heterozygous. So that leaves 4 heterozygous, and then 1, 1, 1, and 1 of all those. Now here's our pairs. We've got a pair, and a pair, and here's another pair. We'll sleuth them out. And then there's another pair. Yeah, don't forget each one of those ones is a 1. And then, we're missing one. Here it is. So there's that last pair. And if you add up all your numbers now, it should come to 16 for your 16 squares. All right, so I'm looking now at kind of the pattern I see, and it turns out it's really fun. This is the waltz. I have my binomial coming true here, right? I've got to match something with big A, something with big A, and something with little a. I've got to match something with big B, big B, little b, and little b, and it turns out I just mesh all the possible combinations. So you can see that happening right here. So there's the 1 sweep of A, 2 sweep of A, 3 sweep of A, and then 1, 2, 3 B, 1, 2, 3 B, 1, 2, 3 B. It's kind of like a waltz with a backbeat. So here's my heterozygous 4, and now we're going to make a little 5-star, and you look out on the sides, and that's my 1 and my 1. And then over here, if I'm my other 1 and my 1. And so now I got that cool little 5-star sitting right there, and now I'm just going to fill in my 2, 2, 2, 2. And so it gives me my 4 2s. So now you can see a 1, 2, 1, 2, 4, 2, 1, 2, 1. A, little a, little a, little a. You can see that right there, and then little a, little a, little a right there. And that's my 1, 2, 3. And then here I've got my 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, and 1, 2, 3, 1, 2, 3, 1, 2, 3. We've got a little waltz going on here. And then if you look at the numbers, it goes a 1, 2, 1, 2, 4, 2, 1, 2, 1. So you can actually, for the rest of your life, do a dihybrid cross by skipping the Punnett square and just building up the binomial waltz. And I think you'll find this to be really useful. You can also here we go. Here's my binomial waltz again. Just let you watch this. All right, so let's talk phenotypes now. We've got our genotypes. That's the types of genes. And your genotype ratio, your 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. So now we're going to look at your phenotypes. That's the physical traits. Well, I guess we don't really know what A and B are, so let's identify those now. A is a yellow pea. Little a is a green pea. B is a round pea, and little b is a wrinkled pea. So let's go backwards. If you look at little a, little b, that is a green wrinkled pea. And think about this. Most of the time when you eat peas, they're green and wrinkled. So it looks like you're eating a whole lot of recessive alleles there. The rest of the peas, let's start with how many of them are actually going to be yellow. Well, anything with a big A. So that's 1 to 2 to 1, 2 to 4 to 2. So that's 1 plus 2 plus 1 is 4, plus 2 is 6, plus 4 is 10, plus 2 more is 12. 12 of your peas are going to be yellow. That leaves only 4, 1, 2, 1, 4 of them that are going to be green. How many of them are going to be round, inflated? That's going to be anything with a big B. So again, that's 12. This time, if you're looking at the grid, we're going across instead of down. Big B, 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 little b, big B, little b, big B, little b. That's 1 to 2 to 1, 2 to 4 to 2. So that leaves just 4 that are going to be wrinkled. Well, how many of them are both yellow and round or inflated? That's anything with a big A and a big B. So that's 1 to 2 to 2 to 4, and that's it. So 1 plus 2 is 3, plus 4 is 7, plus 2 more is 9. So 9/16 are both yellow and round. How many of them are yellow and wrinkled? That's down on the bottom there, 1 and 2. Big A, little b, and big A, little b, little b. Those are yellow and wrinkled. Those are yellow and wrinkled. And then how many of them are green and round? That's going to be little a, big B, and little a, big B, little b. That's also 3. And then we're back to our green wrinkled pea, 1 out of 16. So we have 9 to 3 to 3 to 1. If you're ever looking at shortcutting a dihybrid cross heterozygous problem, think about it this way. If it asks for dominant, that's 9. If it asks for dominant recessive, that's 3. If it asks for recessive dominant, that's 3. And recessive, that's 1. I guess that's it. We're there. I hope that helped. Go Eagles. Talk to you real soon. Bye.
All right, here we go. Now we're going to go over the last questions. Number 4, what is DNA replication and where does it occur in eukaryotic cells? DNA replication is basically the systematic copying of DNA, and it happens in eukaryotic cells. And we're going to learn about DNA replication and the many processes involved in that pretty soon. Number 5, what is DNA transcription and translation, and where does that occur in eukaryotic cells? So we're about to leave the nucleus, right? So DNA transcription and translation is where you make a copy of the sense strand of DNA, the important parts that are going to be read to make a protein. And so the copy is made in something called messenger RNA that's sent out of the nucleus to the ribosome with ribosomal subunits in the rough endoplasmic reticulum and also in the free ribosomes of the cytoplasm. At that point, that messenger RNA goes through the ribosome and it encodes the production of proteins. We're going to learn more about that later. All right, where are we at here? Number 6. What is the eukaryotic endomembrane system and what does it do? Hopefully you learned something about how once that DNA code leaves the nucleus and goes out to the ribosome of the rough endoplasmic reticulum, the code of the ribosomes then basically instructs the order of amino acids. Those amino acids come in on translation RNA and they or in tRNA, and they bond by kicking out of water through dehydration synthesis. It's called a peptide bond. And those amino acids build a long chain of proteins, and those proteins then are going to move from the endoplasmic reticulum to the Golgi apparatus. In the Golgi bodies, they're going to be modified and tagged chemically, and then from there they're going to move into a vesicle, and that vesicle is going to move out to the cell membrane, and it's either going to go in to the cell somewhere, pop open and release that protein, or it's going to release that to the cell membrane itself, where it can be like a transport protein, or it's going to release it out of the cell, and that is going to basically be a secretion, a protein secretion. So either free ribosomes tend to be for the cell when he says, I want this protein for myself. And then endoplasmic reticulum ribosomes, the rough ER, that's going to be for membrane proteins and secreting proteins out of the cell. So the endomembrane system is a system by which the proteins that are built through DNA transcription and translation are then going to be organized and secreted out of the cell. All right. Now, how about functions?
Protein functions. Just check out the Google Classroom Chat Challenge. I want to see 15 awesome protein functions, but let's just go over a couple of them. How about hemoglobin for transporting oxygen, or how about a membrane-bound protein for transporting salts? How about what about a motor protein that causes like muscle flexion and cell movement? One more, an enzyme, my favorite. You've already dealt with those. You know about enzymes. All right, moving on. When you do number 8, you're going to research and describe proteins in terms of their structure based on the monomer building blocks, the amino acids, based on the nature of the bonds between monomers. Remember, amino groups bond to carboxyl groups. They kick out of water. You get an amide and carbonyl at that point, and it sticks the two amino acids together. Primary structure is the order of amino acids in the chain. Secondary structure is the interactions between the amide and carbonyl. Creates like a little twist, a little polar shift. The tertiary structure is basically the interactions of all the R groups. R groups are the variable groups that are adhered to the 20 different amino acids. Their chemical interactions cause the chain to kink and twist and bend and have different properties. And the quaternary structure is where those amino acid complexes bind with other ones to make complete proteins. Protein shape is its destiny. What do you think the quote means? Hopefully you think it means that as you change the shape of proteins, they take on different roles, that the shape of an enzyme will dictate what it can bind with in terms of its the substrate of the active site. The shape of a muscle fiber has to be right if it's going to work as a muscle fiber. And then research and respond, one paragraph only. What is the epigenome and what does it do? Remember, epi means above. Epigenome, above or greater than the genome. The epigenome is chemical alteration to the genes themselves, and it's really important because like an eyeball cell or a skin cell or a liver cell has to know that it's that, and if it was expressing all of its genes, it wouldn't be able to do its job. So to be a liver cell, you have to do liver cell jobs. That means you have to make liver cell proteins. That means you have to have your nerve cell genes switched off and your skin cell genes switched off. The other thing that's really cool about this is the epigenome can actually transfer environmental information beyond from generation to generation, nutritional information, things like that. So if there's a group of animals that are starving, they're going to have a "I'm starving" message in their DNA, and subsequent generations will be able to adapt and respond. So the epigenome is a chemical signaling, chemical triggering that switches genes on and off and changes the outcome of the organism. All right, here's the epigenome in action. Ready? Okay, so now we get to the overarching question. What are the important aspects of a genetics education in the 21st century? Well, we got to start with Gregor Mendel, classical Mendelian genetics, how to run Punnett squares and how to understand the different rules like dominance, incomplete dominance, codominance, multiple alleles. Also understanding polygenic variation, that multiple genes can influence a particular trait. And then we have to get into the concepts of how the nucleus of a cell actually contains the DNA, and that DNA is actually going to have to make copies of itself for mitosis, cell division, so that cells can remain small, so they maintain their surface area to volume relationship, but also so they make copies of each other. And then differentiation, that cells have mechanisms, including the epigenome and genomic information that they have, to basically conduct their program, to make the proteins they need, to run the functions they need, to be the cells they need, be it liver cells, skin cells, muscle cells. We also need to understand how cells make proteins, and that's actually the central dogma of biology. The main idea of all biology in the 21st century is the fact that DNA is the code that is read and transcribed so that proteins can be made at the site of the ribosome, and those proteins can conduct the work of the cell. The last thing then I think that would be really important in understanding biology genetics in the 21st century would be the medical applications and the research applications and the health applications, understanding how these genes basically dictate things that happen to us in our lives, and how things that happen to us from the environment can even communicate with our DNA through the epigenome and give our cells some certain types of adaptations that go beyond just what's encrypted in our DNA code. So hopefully you kind of touched on some of that in your idea of what's necessary for the 21st century. One of the labs that we do is actually imparting a gene into bacteria, so those bacteria will replicate the gene. The production of human insulin for the treatment of diabetes is actually often done by having bacteria with a human gene that's been taken in, and those bacteria go through replications after replications after replications, making copies and copies of the genes, but also actually following the instructions and making human insulin and human insulin and human insulin, which then gets purified and delivered medicinally. So I hope that's useful to you. I hope you were able to complete the first handshake. I'll be doing grading early this week and looking at stuff and helping you out, answering your questions. Let's keep a good thing rolling. See ya.